Okay, everybody. Although Stormy, your face is really small, so I don't know why, but we're live, everyone. Sorry about the delay. We were having some issues with Facebook, but thank you guys all for being on tonight for All Things AS with Amanda. It looks like people are starting to join. Hopefully you're able to find us. I think we had to go onto my personal page to do it live because for some reason it wasn't letting us through ASF. So hopefully you guys will connect with us. But I am super excited because I have two of the smartest, most coolest, most awesome people on the call with me tonight. And, you know, just so you guys know, our Facebook Lives, like we try to keep them within 30 minutes. So it's going to be really hard to do that with you all. But I also know you guys have a life. So we're going to do our best to keep it in that time span tonight. But I'm joined by Becky and Stormy. Words can't express what these ladies mean to me in so many different ways, but I know that none of you care for me to come on and be all sentimental and keep talking because you don't want to hear from me. But the reason why I brought them on tonight, because for someone who does not have a science mind, a research mind, I always am asking Becky and Stormy, especially Stormy lately. When I started, she was like my right-hand woman. I'd call and be like, I have no clue what everyone just said. Can you put it in easy terms for me? And she always would do such a great job doing that. But if we're going to get started, if we can start off by you guys introducing yourselves. So Becky, if you want to go first, and then Stormy.
Sure. I'm Becky Burdine. I am a professor of molecular biology at Princeton University. I'm also the mom to Sophie, who is about to turn 16, which is just hard for me to believe. Who has Angelman syndrome. She's deletion positive and she's doing great. She loves high school. Everything's going well. And also my son, Donovan, who just turned 13 and just said hi.
Yay, I just heard him. Hi, Donovan. Yeah. That's awesome. That's great.
And I've been involved in science and AS since Sophie was diagnosed at 11 months. So I'm happy to be here tonight and happy to be helping the ASF again.
And you have we just announced, I think either last week or the week before, for that you are our new scientific advisory chair. So yay, everyone. Yes. Woohoo. We're so excited.
Big shoes to fill, you know, Carly Williams and then Stormy. And yeah, this is exciting.
You're going to be amazing. Well, Stormy, well, you go ahead and introduce yourself and then we'll just jump right in.
Yeah, so I'm Stormy Chamberlain. I am an associate professor of genetics and genome sciences at UConn Health. I started working on Angelman and Prader-Willi syndrome as a graduate student at the University of Florida with Cammie Brannon and Jim Resnick and Charlie Williams down there. And so I've been at this for a while too. I don't have a child with Angelman syndrome, so I'm just a scientist who works in the area. I study gene regulation and how UBE3A becomes imprinted. That's my bread and butter, and I use human stem cells to do that. And I'm just finishing up my term as the chair of the Scientific Advisory Committee. I've been there for 5 years, I think.
Yeah.
And so I'm happy to pass the baton to Becky because it needs fresh eyes on it.
Well, I will say though, a lot of people were like, "Where's Stormy going? Where's Stormy going?" And I said, "She's not going anywhere. She's still going to be serving on our SAC, so she'll still be around." But you know, we didn't want to. She's given so much of her time and effort to this community, especially to ASF, and so you know, we wanted to give her a break. And so we're excited to have Becky come on.
That was a promise. She promised she was not leaving.
Yes, I know. We were like, you can't go anywhere.
Not going anywhere.
I love it. Okay, so I'm just going to jump into some questions. And what I'm going to do is ask questions based on like questions that I always have. And I would preface this, everyone that's listening, please don't judge me as the CEO of the Angelman Syndrome Foundation that I ask these questions, because you might be like, why don't you know that? Shouldn't you know that? But listen, like I said, no science mind whatsoever. That's why I surround myself with amazing people like these ladies here. But we get a lot of questions at the foundation, a lot around science. And so in research and what it all means. And so my hope is that you guys can answer the questions in a way that you would explain to maybe a fifth grader, because that's kind of how my mind works sometimes. So, and then those of you that are watching, which there's quite a few of you, I'm keeping my eye also on the chat. So feel free to ask questions. We'll try to answer as many as we can tonight. But I thought we would start off first by, right now there's just a lot of exciting things happening within the Angelman syndrome community. It's hard to even keep up with it. Even today, we had a great announcement about the work at the Philpott lab and what they're doing, which we're going to get to in a minute because we've had some questions about what all that means. But I was hoping that maybe we can start off by each of you telling me a little bit about what you're excited about right now for the Angelman syndrome community.
Who goes first?
Okay. Go ahead, Becky.
I am excited the science for sure, and we can talk about what I'm excited about there, but I'm excited that so many people in biotech and pharma are taking a really strong look at Angelman syndrome, and they're excited to be involved in our community. They're excited to reach out to parents and families. A lot of the people I've talked to recently, they'll say, like, what is the thing that's most prevalent in your community that needs to be addressed? And I find this so refreshing because, when we started this journey, when Sophie was 11 months, Art Bodette I know I've told this story to so many people, but Art Bodette told me that night after we got the diagnosis on the phone call, he said, we're going to have clinical trials for this in about 10 years. And at the time, there was nothing on the internet that suggested we were anywhere close to that. Mm-hmm. And I remember thinking, I don't know who this guy is and what he's telling me, but okay. Now I know who he is, but at the time, but he was right. I mean, within 10 years, we started having clinical trials. We started having companies look into this. So I'm so excited about that. And the reason they're doing that is because the science is so exciting right now. Mm-hmm. And I'm excited about the antisense oligos and all of the different companies that are pursuing that type of approach. And the hope is that, one of those really works well at low doses and pays off and no side effects, and our kids, get some kind of boost. And I'm just excited to see how that goes.
Yeah, absolutely. And I'm going to go back to that because I have some questions about the ASOs. And I'm glad that you said the full term because I never can say it. So I just say ASOs. So Stormy, what are you most excited about right now?
I'm most excited that, I'm first of all, most excited that, all of the science that is built up over the years has really paid off. And so,
yes.
Like the different therapeutic approaches are built on solid information, right?
Yeah.
And so it's super exciting to walk into these knowing that they should work, that everything points to this being a really good idea. Both the ASOs and even like the gene therapies, there's been so much refinement of that, and to the point of Ben Philpott's paper that was announced today, that's, people have been working on that. And so these are really carefully thought out therapeutic approaches, and they're becoming optimized. And so I'd have to say that, okay, so I'm excited about that, but we have other shots on goal too, right?
Yeah.
People are talking about things that might even be better than that, or other ways to make it more optimized. And it's really cool to be in this space where, not only can we talk about ASOs and these things that are currently, in clinical trials, but they're already thinking of how could we make it better, right? That's so cool.
Yeah, absolutely.
We're in a great space right now.
Yeah.
Yeah. I mean, I remember when we were diagnosed, 3-4 years ago, there was maybe 3 pharmaceutical companies in the space, maybe a few more, but now I think we're getting close to 30 different pharmaceutical companies in the space, which is, which means that there's something here, right? That there's something that industry and pharmaceutical companies think that they could really support and help out, which is exciting.
And I don't think we should leave. I mean, the science is solid, as Stormy says. We've got a solid scientific base, but I don't think we should overlook the fact that we have a lot of other things going for us as a community. We are a small community, but we're pretty connected. Mm-hmm. We have a lot of people working for us. Everybody's been really willing and able to jump in and talk to companies when they want to be talked to. Yeah. People have been participating in trials. They've been following the rules of the trials. I think, everything that we get involved in shows other companies that this is a community that's worth supporting, that we're going to be there to help them.
That's a great point, Becky. I think it's great to be able to reach out to families from the foundation's perspective and say, hey, I need 5 or 6 families that want to kind of give a parent perspective, and everyone's like, me. And I love that. It's wonderful.
Right. Well, we
And
have the clinics,
the
we have the
database,
database, we have the
databases.
clinics,
Yeah.
the natural history study. I mean, like,
Yeah.
you know, if you look at what they tell rare diseases to do so that they're ready for this, you know, we're there.
Yeah.
So we're in a good position.
And they point to us, right? Everybody points
Yeah.
to Angelman Syndrome Foundation now and in our space and says, this is what you need to be doing for your disorder. So it's good to blaze those paths for others as well.
Now, we still have work to do. Don't get me
Yes.
wrong. We're not resting on laurels here. We got work to do.
Yeah,
But
absolutely.
we're in a good position. And the fact that all these companies are looking at our community says that's true.
So can one of you explain to me in very simple terms what the ASO is hoping to do? And is this the cure? Like, is this the thing that will cure Angelman? Or help me understand what the ASO is actually going to do. Whoever wants to go.
So this is what I study actually, is the gene regulation. And so the ASOs, what they are is they're short little DNA/RNA pieces. And they sit down on the RNA that is the antisense transcript. So UBE3A is a really peculiar gene in that UBE3A protein is made by mom's copy of the gene, the copy of the gene inherited from mom in neurons, and dad's copy is shut down by this RNA called the antisense transcript. And they, like a train, two trains on the same track, they crash. The ASO cuts the antisense train and kicks it off the track, letting dad's copy of UBE3A be produced. So since your kids with Angelman syndrome either are missing mom's copy of the gene or have a broken maternal copy, dad's copy is sitting there ready to be turned on. And so these ASOs turn it on. Is it a cure? A cure is a loaded word, right? It puts the protein back that your kids are missing, but that protein was required during development, and we really don't understand when it, we kind of understand when it first comes on, when it's first used, but we don't understand how important it is early on versus later on. But thanks to some mouse models and some work from E.P.L. Gersma's lab, we have a pretty good sense that there will be benefit. Does that amount to a cure? I suppose it depends on your definition of a cure. I think there's going to be benefit and probably really good benefit for many people. So I'm very optimistic about that.
Yeah.
Yeah, I think cure is a loaded word. I mean, but, you know, if we think about cure in terms of being free of symptoms, you know, because, you know, are our kids never going to have had Angelman syndrome? No. I guess you could think of it this way. If you've ever had chicken pox, which hopefully most of you haven't, but if you are, you know, my age or older, you might have had chicken pox. I've been cured of chicken pox. I don't have chicken pox. I still have the virus. Like, that virus is never going away, and hopefully I don't get shingles. But have I been cured of chicken pox? Yes, but no, right? I think the thing I always think about is this. Our kids, as all of you know, have this really delayed ability to learn. And, you know, it takes a long time for them to be able to learn something. I love how this is happening right now, because, like, we're all living our lives and Amanda's running off the Instagram. And, you know, our kids, they can take a really long time, but then they learn something and they keep it. And part of that is because the neurons aren't functioning correctly. And whether you look at Ipa's work with the mouse, and we're not mice, so it's sort of hard to, like, you know, do a one-on-one there, but even in the mouse, whenever they turned on the paternal UBE3A in those mice, or the copy they added, the mice learning and memory was restored. And, you know, if my daughter could get her learning and memory back up to speed, and then we could start teaching her more things and she could acquire more skills quickly, that's a win.
Yeah. I think that's a good, I mean, I think that's a good example and a definition of, you know, I think the cure means something different to everyone, but hopefully, you know, for me as a parent, I just want the best quality of life for Jackson. And if that means that some of these symptoms are fixed, and if this ASO can do that, that would be huge. Right. So I have heard people use the term, like, an ASO is kind of almost like a Band-Aid until we get to the true, like, transformational gene therapy, which, and once again, these are just my mind, this like question I'm asking. what is true gene therapy? Is that when we start talking about like CRISPR? Because with an ASO, you have to have that. We don't know how many times you have to have that. The individual will have to have an injection, right? But tell me, like, if someone can explain a little bit about some of the terms we've heard about CRISPR, about the snRNA, about AAV, like those different methods that are being looked at right now. If you guys want to tag team that, that would be great in very simple terms.
Sure. So the CRISPR approach that's been talked about, the AAV gene therapy that was the subject of Ben Philpott's paper, and ASOs, they all do essentially the same thing. They put the gene back.
Okay.
So they all put the gene back in, but in slightly different ways.
Okay.
And each of those have their own benefits and their own drawbacks. And so I'm not 100% sure that the other therapies are quote unquote better, but it's another shot on goal in case one falls short. And so, and it may very well be that where the ASOs fall short, one of these other approaches pick up, right?
Yeah.
And unfortunately, we really can't know this until they're thoroughly tested. These are difficult experiments, and so I'm forever grateful for people who volunteer for clinical trials because you're brave, and we need that in order to understand how to go back and do better. We keep doing better. We keep trying to do better anyways, but
yeah.
Thank you guys for participating.
I think what I like about the antisense oligo approach is that, like Stormy explained, we stop that train so that the gene that's already there can do what it normally would do, right? So it's sort of a very small, and it's not that invasive, I guess is what I'm saying. And we know that genes have bizarre ways that they regulate themselves, and there's lots of labs that study these things. But I think if we could just let the gene turn itself on, a lot of the things in neurons would sort of maybe take care for themselves.
Yeah.
I hope. So that seems the least problematic. I mean, some of the other approaches, when we talk about pros and cons, it's easier to see some of the pros and cons. But what's important is that we push on everything, because we don't know what's going to work until it works. And if we stopped right here and said, ASOs, that's all we need to worry about, we're done, we could really end up hurting ourselves, because maybe that's not the way it's going to be gone. Or something, I guess it's okay to say this, like, because I know Stormy's involved in this research, another idea would be to use something like AAV, which is it's a virus that's been modified so it's not damaging in any way, but it does go in and insert in your genome, and then it's permanently there. So it's sort of like having chickenpox. That virus is going to be there forever once you insert it. And maybe what we could do is have the virus carry the antisense oligo so it would stop the train.
Oh, okay.
And then it would be permanent, and the train would be stopped, and the control would be taken over. That seems really promising to me, but we have a lot. I mean, it's Stormy's work, but we have a lot of work to do before that becomes a viable option. But
that's the idea, though, is that we're building on ASOs, right? So each of these build on something that we already know. So it's really great that the ASOs are in clinical trials. When we see how well they work, we will kind of know the things that they're great at and the things that they're maybe not as great at. And we try to fill those gaps. And as research, the SAC, we try to support the studies that fill those gaps in before we even know that there's a gap, right? That's
before we know we need them.
Yeah. We have to do that work now to make sure that we're ready for it when we learn about the gaps.
So there's an interesting question, I think, that I'll ask you guys from the chat, because I think it's a good time to insert it. Someone asked, are there any other crucial genes other than UBE3A that are missing in the deletion genotype that create noticeable deficit for those with AS?
This is such a great question.
Right?
Desiree. And it's a yes and a no. So it's a no in that the only gene that causes Angelman syndrome is UBE3A. And we know that because of mutations in UBE3A or kids with UPD or imprinting disorders. Like, that's the gene that really matters. But we also know that deletion does cause other issues, because the kids with deletions tend to be affected more strongly by Angelman syndrome. There's like one or two other genes that are imprinted in that region, but they don't seem to be important. What might be important are the receptors that happen to be in the deletion region. These are called GABA receptors, and they're very important for how neurons work. And no one's really done any research to see if having half the amount of GABA receptor encoding regions in DNA is problematic. We assume it must be because of the fact that our deletion kids are are affected more strongly. That's not 100% across the board, but in general. But in terms of what do we need to fix or restore for correcting Angelman syndrome, it's just the one gene, which is, again, why all the biochem and pharma companies are like, this is a good one. Right.
And to a point we made earlier, we funded studies to ask this question carefully, right? So we funded Ipel Gersma to make a mouse model to model the deletion, and his lab would be great for this because they've already done a lot of phenotyping, studying how the mouse behaves with just UBE3A mutated. So now what happens when it's UBE3A plus these genes or just these other genes alone? And so we've funded that study, and then we've also funded a study. HERC2 is another important gene in the region, and we funded a structure study with Gilles Travet to ask how it interacts with UBE3A, because it could be that those two work together, and in that way, HERC2 might be important.
Yeah, the fascinating thing of being able to sit on SAC, which is Scientific Advisory Committee meetings, is that sometimes there is a lot that I feel like we're still trying to learn, right? And some of these things that, may not seem super sexy or appealing, like that we're funding, it's some of it's really, really important for us to be able to understand. I was hoping that you could we get this question a lot, especially lately, if you can give some hope to the families. The question is, will there be any clinical trials coming up for mutation or UPD? Can you talk about that a little bit?
So I think we've talked about this before, and I'm happy to do another webinar on it at some point if people are interested, about what goes into thinking about planning a clinical trial. Not from a pharma perspective, but just from the perspective that I've learned sitting through some of the planning that happened, and talking to the FDAs and things like that. And the most important thing with a clinical trial is you want all of the people in that clinical trial to be as similar as they can possibly be. And you can't, right? You can't make them all similar. You'd love to take one person and clone them 100 times and then do your test, but you can't. We have more individuals with deletions. So when we're trying to make that population as similar as possible, they're going to go for something like where they can get the most kids. And again, the kids with deletions tend to be more strongly affected. So if you're looking for something to show improvement, you can, if you're starting down here and the improvement's here, that's easy to see. If you're starting here and the improvement's here, it's a little harder to see. So it must be really frustrating, and I completely understand it. I'm frustrated because my daughter had spinal fusion surgery, which rules her out for almost all of the trials that are going on right now, and it's killing me because this is what I worked for forever. But we're just going to have to be patient, because if a trial does show an effect, they're going to have to open it up to more people. They're going to want to see how it works in mutations. One of the good things about the oligo trials is UPD. We worry that we've got 2 copies that might turn on and maybe too much UBE3A is a problem. Well, with oligos, we can titrate it, right? So they'll have to do those kinds of tests where they maybe see if less would be good for a UPD kid. Though they're coming, right? If we see positive effects in some of these first rounds, I'm sure they're going to open it up to UPD and mutation. So it's hard, but just be patient.
Yeah, you're right. Stormy, you want to add at all?
Oh, I just, we made stem cells from UPD patients, and we actually tested ASOs in those. And so that's in a bioarchive. It's freely available to see how well they work. And to Becky's point, we use a lower dose of ASO to get a good effect. And so, I love Becky's suggestion to be patient, because when it gets to you, the good news is that you'll probably use a lower dose, and that probably comes with its own benefits too.
Interesting. I feel like I'm learning so much, by the way. So I know we're a little over time. Do you guys mind staying on just a few more minutes since we got started late? Okay, good. Okay, so I have my list of questions, and please feel free. I know there's a lot of people watching, and feel free to ask some questions. So when we posted today about Ben's article, there was a lot of questions about what this meant. Stormy, or I don't know if Becky, you had time to read it. Can anyone just kind of help us understand the excitement about that article today?
I think Stormy's probably in the best position to do that.
So Ben's article is kind of exciting because, as if UBE3A wasn't complicated enough.
Yeah.
In humans, there are 3 different versions of the protein, right? And so not only does it make UBE3A, it makes a short and a medium and a long form of UBE3A. And we really don't know whether it matters which size of UBE3A it is. So what Ben's work did was they found kind of a clever way to make the different sizes of UBE3A from one gene therapy molecule. So what that gene therapy molecule means is that he took the gene, put it into this AAV vector, like Becky had explained before. It's a relatively harmless virus that you strip all the stuff inside out of it and then use it as a Trojan horse to deliver the gene to your neurons. So they did this in mouse and tested how well that works to put UBE3A back. And kind of what's cool is you get the different sizes of UBE3A, which is very cool, right? And so again, it's an improvement on the idea of gene therapy with because we've known for a while that there are 3 different versions of the protein, and we want to make sure we don't miss something by simply committing to one version of the protein and only sticking that in. So that's what's super exciting about that paper.
Yeah. All of this is just super exciting, period. I'm sure for individuals like you and Ben and Mark and David Siegel and other people who are working in the space to start seeing some of your work like come to life and be published, and now seeing it actually coming into clinical trials, as a researcher, it's got to be incredibly thrilling for you.
It's very fun to know that you make a difference, right? Like most of the time, those of us who love science, we went into this because we love science, and we go into our labs and do our little experiments. But your guys' community provides an opportunity for us to have a sense of purpose, right? And so it's amazing how rewarding that is. And I tell my students this every day, and I think it motivates them to do the work for the right reasons, right? So you guys give us such a gift by letting us use our science for good things.
Well, I will tell you, when I first was diagnosed, like I was doing some research and reading, and I felt like the name Stormy was almost like talking about Brad Pitt of like the world, like, you know, like the celebrity of AS. And so when I first met you, I was so like shy. I was like, oh my gosh, it's Stormy Chamberlain. Like, I know. So I think you've done so many amazing things for our community. So anyway, I got a question, 2 of them, but I'm gonna address the other one in just a minute, Lauren. But Natalie asked the question of, have there been any studies to show what % of UBE3A is needed to see improvement? Thinking of kids with Mosaic genotype who have verbal speech.
I don't think there's been one that's been done that sort of directly addresses that, but it could be done if I think about it. But you're absolutely right, Natalie, because I have met some kids that are Mosaic that blew my mind.
Yeah, right.
Yeah, because the first kid I met that was Mosaic, I saw her with another little boy, and they were having a discussion. And they were both about 4, I guess, and he had a donut. And she pointed to him and she said, "Where?" And he said, "My dad gave it to me." And she said, "Dad?" And he said, "Yes." And she's like, "I want." And he said, "Okay." And he took her hand and they walked off. And then someone said, "That's my daughter with Angelman syndrome." And I'm like, "Excuse me?"
Yeah, you're like, no.
It blew my mind.
Yeah.
And she's amazing. She's an amazing kid, but that was one of those things where I'm like, oh, we don't need a lot, right? We may not need a lot. We may only need some neurons to actually do their jobs. Maybe it's specific ones, maybe, and there are studies where we're trying to figure that out, like which neurons have to have it. Do they all have to have it? Some of them have to have it.
Interesting.
Are some regions important in the brain and others not? We just don't know. There's so many good questions in the chat.
I know, I'm trying to keep up as well.
I don't know, but Natalie, it's a great question. And I think that's, we can think about that on the SAC.
There's one paper that gives a hint. Wenhan Ten published a paper about 2 families that have a mutation in UBE3A. So these families have a mutation in UBE3A, and it disrupts one of those forms, one of those isoforms of UBE3A, the short form. And subsequent work in Elgarisma's lab and in my lab showed that if you make that same mutation in human stem cell neurons or in mouse neurons, it results in leaving only about 15 to 20% of UBE3A protein. Those kids speak in phrases and they have improved motor skills. And so
Wow.
in my mind, I use that bar as we know that there would be substantial improvement if we could put back 15 to 20%. And that's, based on those papers together. I think that's a good target.
Yeah.
So I think we should
go ahead.
I think we should go back and answer Lauren's question because it's been.
I was going to answer that really quick. I think
Yeah.
the question is, should we be saving now for when a treatment does become available? It's scary to think that we're getting so excited about all these potential treatments, yet when it becomes available, can we afford it? And I think that's a great question, Lauren. And the good news is that we've been thinking about that at the ASF for a while and already working on what this looks like. And a couple of things we're doing. We are partnering with FAST to come up with the burden, like a cost burden of disease paper, because it's going to be really important for us to be able to articulate and convince, insurance companies, Medicaid, whoever, that it's going to cost them less to help fund these treatments now than to fund the care of an individual with Angelman syndrome over the lifetime. So we are working on that stuff now, and we're already talking with insurance companies and Medicaid so we can advocate. That's why we do work on the Hill to make sure that when these drugs come to the market, we're able to provide whatever resources we can to the community to make this accessible to all, because that is very, very important to the Angelman Syndrome Foundation, that anyone that wants this treatment, that we can help them get access to it. And we're also working with the Assistant Fund, which is a huge fund out of DC that creates funds for different disease areas. And what happens is usually when there is a pharmaceutical company that is in phase 3 trials and it's looking good, they'll partner with an advocacy organization and create an assistant fund. And this fund can, they'll put money into it. It can grow. Families can request access to dollars to it if they need to help get their access to these treatments. So long story short, we're thinking about that. Daily it keeps me up of thinking, like, how do we make sure people have access to this if they want it? And so we are thinking about that and working very hard on that right now.
And, and as a community, it's really important that we support and send people to represent us in government, people that are going to make it affordable for everyone to have healthcare or access to Medicaid. We don't want them cutting our programs.
Yep.
Like, we need these things. And pharma and biotech know that if they price this out of the realm of possibility, then they're not going to sell it. And then that's not a business decision that makes it for them. So I know it's really scary to think that's going to be there and you can't afford it. I wish I could reassure you that we're going to do everything we can do to make that not happen.
Well, and we're going to partner with anyone we can to make it happen. Bringing these organizations together, bringing FAST, ASF, bringing CAS, bringing all of the Angelman groups together to say, listen, we're going to work together hard to make sure that we are able to give, to get access. We commit to you that will happen for sure.
Absolutely.
So there are a couple other questions I want to get to, and like I said, I could talk to you guys all night. So one of the questions, I think this is a question that comes up a lot, and Sammy, I will connect with you. The question is, I have twins, each diagnosed with AS. How can I help? I'm going to connect with you on that. But really quickly, I want to address this question because it comes up a lot. The question is, my daughter's 28 years old, diagnosed at age 2. Will any of this research help her? I've heard so much of this before. Not much has helped her yet. It's something that comes up a lot. This question comes up constantly, and I would love for your guys' feedback on this.
So I think, I can make a really big long answer, but I'll do something a little bit shorter. Those of you who have adults with Angelman syndrome, and you've paved the way for the rest of us, right? And like people in my generation of AS have paved the way for the people who are coming right now. And it's really important for us to make sure that we don't leave anybody behind. And I think the work that we talked about earlier, especially with thinking about what can be restored even in adult mice And those adult mice are adults, like they would be like 30, 40-year-old people. We could still restore learning and memory. And that means that some of these treatments, if they do work, they'll work for everybody. They may not, restore, say, motor functions, because if you've learned to walk in funny ways, then your muscles and your bones and everything have sort of adjusted to walk in funny ways. You're not going to immediately get a treatment that restores UBE3A and suddenly walk like you've never had a problem before, right? Some of those things are learned, and some of them are, sort of ingrained in how things work. But if you can improve learning and memory, then, the sky's the limit. We learn all of our lives. I could go out right now and, start trying to pick up French, and I could do it. And that's because your neurons never stop learning. So if we could if we could restore that for our adult individuals in the community with Angelman syndrome, they would see some improvements. We're not going to know till we know. But yeah, they're not going to be left behind. This is not going to just be for kids under 1 and the rest of us have to, stand on the sidelines and be sad. I think it's going to be for everybody.
Stormy?
And on the SAC side, we continue to look for ways to help older individuals with Angelman syndrome. And so one of the studies we funded most recently was from Robert Carson's group, looking at tremor, myoclonic tremor. And if I understand correctly, that affects older individuals with Angelman syndrome specifically, but to try to understand what exactly is it, and what are the best treatments that work for it. And what's really cool about that study is he's using experts in movement disorders to try to understand it better, right? And from an outsider perspective a little bit. So instead of just assuming we know UBE3 and assuming we know Angelman syndrome, he's fishing for, well, what can other professionals tell us that might inform how we better understand myoclonic tremor? And so we keep looking for ways on the SAC to invest in research that would be more helpful to older individuals. And it's of course easier to do this now that ASOs, and we have some things in the pipeline for clinical trials. It makes it easier for people to go back and say, now what else can we do to help? And we haven't forgotten about it.
Well, and I think the other thing, and I think Becky, you bring up a good point, because I think it's important for people to know, from what I understand, like, let's say that your child or your individual with Angelman syndrome does get an ASO, continuing doing therapy and learning, because it's not like it's going to happen overnight. Like, it's going to take time to like relearn. So getting, continuing to work with your therapies, continuing to work with your school, like it will hopefully grow and develop. Is that correct?
That would be the hope. I mean, keep in mind too that, and Ipo will tell you this when he talks about his mice studies and stuff, when we do a treatment in the mouse, we do the treatment and then we throw the mouse at a test and we say, do you do better than if we threw a mouse who didn't get the treatment at the test? That's not the same as one of our kids getting a treatment and then continuing with physical therapy and maybe making strides quicker or better, or doing OT and suddenly their point gets better. Like, we don't give the mice time to sort of train and learn things and then test them. We test them right off the bat. Did you get a benefit or not? That's not how clinical trials work, and that's not how humans work. No. So the hope would be that a treatment, if it does have a benefit, you're going to see that benefit sort of maximized, with therapies and ongoing treatments and ongoing things.
That's interesting.
Very different.
So, okay. Yeah, really quickly, so someone asked after this talk, where can you find papers? So some of the stuff that you've talked about, Stormy, how can you best kind of give guidance to the community if they want to learn? I mean, because I will tell you, like, there's a lot of stuff out there. We actually cataloged all the papers that are out there, and it's over like 3,000 or 4,000. So there's a lot. So can you give some suggestions to people on like, where after this talk can we find papers and how to go about doing that?
Yeah, so
because it's a lot.
It's a little tough. I mean, the place scientists look for papers is PubMed. And you can type in a search term, Angelman syndrome or UBE3A and get updated papers. Some of them are behind a paywall. But on the down low, if you bother one of us scientists,
good to know.
We can get them to you.
Yeah.
In a lot of cases, though, they're publicly available anyways. There's a lot of free stuff out there too. The other, I think the other cool thing to do is to Google, to be honest. A lot of these papers will come with a press release that explain it in roughly understandable terms.
Yeah.
And it's the abbreviated version. And so you'll get the take-home message from the press release too. And so that's a good way to just stay on top of the news at a high level way. But I think I forgot to look whether Ben's paper is freely available or not.
Yeah, I didn't look either. I got so excited about it. I didn't see. But you can also, just so you know, on our website, if you go to ASF funded research, you can literally, it's like a scroll down of all we've funded. And if you click on it, it'll give you an explanation of that funding. And if there's a paper that's been attached to it, usually that paper will be there in hyperlink for you to view. If we're able to put it on our website. Yeah.
And on some of the Facebook groups, I know in the epilepsy group and the research group, a lot of times some of these really classic papers will have been uploaded at some point. If you go to the files sections of those groups, a lot of times these papers are there.
So Ben's paper is available according to Angelman Syndrome Foundation, it is available. So that's Catherine. So thank you in the background, Catherine, for doing that. So let's really quickly, if we could answer Stephanie's question, then I'll wrap up because I know I've taken a lot of your time.
I'm not hearing any real answers. I guess I don't understand, when will any of this research help her? Yeah, Robert, I think it will. I mean, maybe we could discuss offline.
Yeah. How about Robert, I'll connect you with Becky and then you guys can talk. Sorry if you don't feel like we're giving real answers here, but we'll definitely connect you so we can have more of a conversation with you. But let's get to Stephanie's question. How do we help support these future measures at a local clinical level, advocating for up and coming treatments paired with continued therapies, especially on rural areas where physicians are still seeing ASSCP or autism? Great question. Who wants to take a stab at it?
Well, I mean, maybe you should mention that, we do have these committees, right? We have the advocacy committees and things like that. And Stephanie, you'd be awesome on some of these committees just because of of all of the skill sets you have. I think what we have to do, I mean, the clinics are great, but we need to be able to get, care out to everybody, and not everybody can get to a clinic. And I think we need to the clinics are starting to do this. They're starting to put together papers on care, right? We're trying to make sure that people publish, like T-Bert publishing his Valium Reset is great because you can walk up to a doctor with a paper and say, this is what we need to do. Right. And they listen to that, right? They don't listen to, this mom on Facebook told me this. They're like, yeah, whatever. But a paper, somehow that, we know that makes up for that. Sure. So I think that's one of the things we can do as an organization, is making sure that we have those position papers available from clinicians so that we can make sure that everybody has the information they need to properly treat Angelman syndrome.
Yeah.
Go ahead.
No, I would say that we're going to be moving into like a really aggressive strategic plan when it comes to our clinic network, because we know these clinics are going to be vital, not only for care long-term, but also for clinical trials. As more and more come on, they're going to be happening at these clinics. So we know that getting to a clinic sometimes is just not an option, or it's something you would rather work with your team back home, which is totally fine. So we're going to be aggressively putting out a lot more standard of care papers, one-pagers on different symptoms that you can take and you can share with your clinicians at home as much as possible. So if you want to understand more about what we're going to be doing with the clinical landscape, you can check that out on our website. But we are going to be working really, really hard in that area coming up. So,
Robert, let me just say this, because I think what you're expressing is not uncommon. Yeah. In the community, and because we have been hearing this a long time. I've been hearing it a long time. And things like that. But I think it's hard sometimes to realize that we actually have made huge strides, right? And since Sophie was diagnosed, how we treat seizures in AS has changed completely. To the point that Dr. T-Bert didn't have any AS patients that qualified to be in the trials for Epidiolex because everybody was well controlled. That's amazing if you think about it, because seizures is one of those things that we're so scared about. We know a little bit more about how to at least control constipation in AS. We know what the good treatments are. We actually have a lot of treatments for Angelman syndrome. They're not cures, But, you know, a lot of the symptoms that we deal with in Angelman syndrome, we do have treatments for, and we have ways of controlling those symptoms. And I know it's so hard to understand this, but, you know, we cured the mouse model of AS in 1998, and, you know, it's 20 years later. That's really fast. I know it doesn't seem like it, but in terms of trying to go from just getting something to work in a mouse to having clinical trials in humans, that's an unheard of speed. And especially for a community, a science community that for such a long time did not have the resources and funding it needed to move stuff forward. And, you know, that's the other reason why it's amazing how quickly pharma has taken us up, because we have managed to go along without, you know, the millions and millions of dollars we really needed 20 years ago. So I get your frustration. I really do get it, but we actually have made a lot of progress. And I still think the treatments that are in clinical trials right now could potentially help your child.
Yeah, I think, and I feel like I don't really have room to talk here because my child with Angelman syndrome is still young. But the one thing I've learned from being in this position is I feel like it's very important for people to understand the path that has been laid for individuals like me and my child who are younger, from these families who have worked their butts off for so many years, whether it's fundraising, whether it's, volunteering at one of the organ foundations, whatever it may be. Like, the thinking back on where we are now and who got us here, it's got to be very challenging to be in a community for so long when, the promise keeps coming of like, it's coming. And the days, the years keep ticking on, and you keep seeing your child struggle. Like, I can't even fathom or pretend to understand that frustration, but I hope that everyone that's listening knows, like, our goal really is to continue to research and find ways to treat, but also to support you on that day-to-day journey, knowing that it has been a long time and for some people, but what if we can alleviate something, whether it's to get you an adaptive bike, whether it's to get you, a meeting with an IEP, whatever it may be, how we can help you through the, the day-to-day struggles is something that we're really passionate about as well. So, I don't know if I have much more to say, but I do think about you guys on a daily basis when I think about the path that you've laid for our community. Stormy, did you want to say something?
Oh, I just say I sympathize as a researcher. Yeah. We always wish we could do more, right?
And faster.
It's equally frustrating on our end that we can't move it faster. Unfortunately, we ask good questions, and sometimes the answer wasn't right. We didn't get it right. We fail. We do, and that's part of research. Thankfully, enough things have worked out that we have some shots on goal in clinical trials. I agree with Becky. I think they probably will be of some benefit to your individual, your loved one with Angelman syndrome, and hang in there. We're still trying.
Yeah, I appreciate that. So we could keep asking questions, but
we can answer the last one.
Okay, go ahead. Yeah, go ahead.
Have any other genetic syndromes been successfully cured? And I like how cured isn't in the thing. There have been successful gene therapies for various different things, and more of them are coming every day. Some of the, I think probably the one that would be most helpful to point out, although Stormy can come up with other ones probably, is for spinal muscular atrophy, right? Because this is a devastating illness. And one of the forms, it's usually fatal before the children are 1. And they have been doing, various different gene therapies for SMA, and these kids are actually surviving, and they're making progress, and they're meeting their milestones. And you can imagine for those parents how relieving it is to know you're not going to lose your kid before they reach a year. I can't even imagine. That's been good. There are some genetic forms of blindness that, we're going in now and trying to repair. They're doing one eye at a time versus the other eye, and they're seeing some benefits. People are starting to get some sight back that they had lost. And, you know, this is a silly thing, but this has been going on for a long time. There are some genetic diseases of the blood that can be cured with a bone marrow transplant. We've been doing that for a long time.
Right. Interesting.
So, yeah.
I think for neurodevelopmental disorders, we're trailblazers. Like,
yeah,
there are disorders that have probably had more money pumped into them, have been more heavily studied, have more researchers chasing it, and we're further along than they are. So I think we're trailblazers for neurodevelopmental disorders, though. And so that's a blessing and a curse, right? It's fun to be cursed, but makes it hard to know what to expect.
Yeah, absolutely. So I will, if we could do one more, just because I think I get this question a lot of people are just so excited to hear the results of the ASOs already. And I think that, you know, people are just nipping at the bits of saying, like, you know, what are people seeing? Like, tell me more. Can you guys explain a little bit, like, as much as we'd love to share, you know, as much as pharma would probably love to share, like, what they're seeing, maybe explain a little bit of when we might be able to hear some of the results of these studies, or these first phase 1/2 trials?
Well, for pharma and biotech, I think it's important to keep in mind there's a lot of regulation, around when they're allowed to share and what they're allowed to share. You know, it's actually a huge violation for someone to sort of whisper to you, you know, like, hey, this is what we're seeing. Like, that shouldn't happen. And it has a lot to do with, you know, bias and moving the needles and improperly influencing things. And, you know, those rules are there for very good reasons. So they tell us when they tell us. And we've been pretty fortunate so far that companies that are working in this space, when they have a result and they're at a point where they're allowed to share it, they do. You know, and you know, I did some consultation for Ovid, but I was very happy to see that Ovid put papers out as soon as they could. They told us what the results were as soon as they could. When the second follow-up trial, when they unblinded it and saw that the results were not what they hoped, they told us immediately, right? You know, there are clinical trials on clinicaltrials.gov that have been going on for 20, 30 years, and no one's ever seen a result because the companies just went, you know, and it's a big frustration.
Yeah. So, I mean, I always tell families, like, really, no news is probably good news at this point, right? Like, I mean,
correct. Because if there's bad news, they have to tell you.
Yeah, exactly.
Right.
And they have to stop the trial, right?
It's just frustrating.
Yeah.
And Stormy, what did you say? I'm sorry.
Oh, I said, and they have to stop the trial. If there's bad news, they have to stop the trial, right? It's unethical to move forward if they know that there's these phase 1 clinical trials are safety trials.
They're safety trials. Right.
If there's a safety issue, they have to stop it. And the FDA is getting better about cracking down on reporting results as well. So
absolutely.
You know, it's not a good substitute. I mean, I know you guys want to know everybody. I want to know, right?
Right.
I want to know too. And I'm sure that this is happening. And, you know, I'm not saying judgment or anything like that. But one of the reasons the companies say to you, please don't share results with other people. Please don't get on Facebook and make private groups where you talk about what's happening, is because it's so easy to influence how you feel about things and what you think is happening, and you can't stop it. It's not something that you can control. Like, we have all kinds of studies showing that the ability of your brain to modify what you think you're seeing is completely out of your control. But, you know, if I'm in a trial and we get in a group and you tell me, wow, my kid's really starting to improve their fine motor skills, and I'm like, maybe my kid would improve their fine motor skills now that they're in the trial. And so I up the OT that my kid's getting. And suddenly my kid's showing better fine motor skills. And that's great, but my kid's on placebo. And I've just pushed the needle and I've made that noise to signal ratio go like this. There are reasons why we, and especially when our population is so small and we're so rare, like, we have to do everything we can to make these trials perfect. Like perfect. Because if there's a signal, we want to see it. We want the FDA to see it, and we want the FDA to say, this is amazing, give it to everybody right now.
Exactly. Well, I mean, I think that's hard, especially for a parent, of like, I want to know, right?
I want to know right now.
I just keep telling people, like, no news is good news. So if you're not hearing anything, that, like, just, even though it's frustrating, just count on that as being good for right now. But as soon as they can tell us, they will tell us, right?
Yeah, and we'll hound them until they do, right?
Exactly.
There's too many of us that are like,
yeah, don't think we don't talk to the pharma companies on a weekly basis of like, what's going on? Tell us more. What can we tell our community? So you definitely have advocates in that way. Well, ladies, I went way over time tonight. I think we all three could be on the phone. We might have to do a part 2, just so you guys know. But thank you guys for being on tonight and for all your dedication, support, and knowledge for the community. Hopefully I'm not speaking out of turn, but I hope everyone that's listening also knows that these ladies love talking to families. So if at any point you just have questions and want to talk more, let us know. We'd love to connect you to a researcher, and they'll share what they can, and they'll talk how they can. But you guys are amazing. Stormy and Becky, thank you so much. Go back and have a great weekend. And for all of you guys that are watching, thank you all for what you do for the community, especially today with the announcement with Ben's work. None of that would have happened without ASF funding, and none of that would have happened without the community working really hard to bring funds in. So thank you guys for all that you do. Have a great weekend, and we'll see you at the next All About AS with Amanda. Have a good weekend, everybody. Bye.